Okay, so what is a genetic code and how to make a protein? I will discuss it with you in this video. Have you ever written a secret message to one of your friends? If so, you may have used a code to keep the message hidden. You might have done this also in internet. For instance, you may have replaced the letters of the word with numbers or symbols following a particular set of rules in order to, in order for your friend to understand the message, they will need to know the code and apply the same set of rules in reverse to decode it. Encoding and decoding, this is the process. So decoding messages is also a key step in gene expression, in which information from a gene is read out to build a protein. So in this video, I will take a closer look at the genetic code, which allows DNA and RNA sequences to be decoded into the amino acids of a protein. So as a background, genes that provide instructions for proteins are expressed in a two-step process. And in transcription, the first one, the DNA sequence of a gene is rewritten in RNA. In eukaryotes, the RNA must go through additional processing steps to become a messenger RNA or mRNA. In translation, the sequence of nucleotides in the mRNA is translated into a sequence of amino acids in a polypeptide and on a polypeptide or a protein chain. If this is a new concept for you, you may want to learn by watching the previous videos done by me. All right, so what about codons? Cells decode mRNAs by reading their nucleotides in groups of 3 called codons. Okay, these 3 group nucleotide bases are called codons. Most codons specify an amino acid, and like this, 3 stop codons mark the end of a protein. For example, this one, this is a stop codon. This is not translated into an amino acid. This is just to say that you have to stop here to produce amino acids or chains of amino acids. And 1 start codon, this is start codon, adenine, uracil, and guanine. Uracil is counterpart of thymine in RNA. Instead of thymine in RNA, they use uracil. 1 start codon, AUG, adenine, uracil, guanine, marks the beginning of a protein and also encodes the amino acid methionine. Okay, so it starts at here, but it also functions to produce the first amino acid, which is called methionine, and usually showed as these 3 letters for short, MET, met. Okay. Codons in an mRNA are read during translation, beginning with a start codon and continuing until a stop codon is reached. And mRNA codons are read from 5' to 3' in this order, in this direction, okay? From 5' to 3'. And they specify the order of amino acids in a protein from N terminus or methionine to C terminus. Okay. All right. Well, let's check these code tables for in translating these three codons into amino acids, this is the chart, okay, that you are going to use how to translate these three sets of codons. Sorry, say sets of codons or three ladders of nucleotides. The full set of relationships between the codons and amino acids or stop signals is called the genetic code. Okay, and the genetic code is often summarized in this table. Notice that many amino acids are represented in the table by more than one codon. For example, this one is produced by either this set of bases or this codon set or this codon set. You know, they are different, but they produce the same amino acid. Okay, for instance, there are 6 different ways to write. Where is it? Leucine in the language of mRNA, and leucine is this one, 4 of the leucine over here, and 2 of them over here. So all these 6 codons produces the same amino acid. Okay. An important point about the genetic code is that it's universal. That is, with the minor exceptions, virtually all species from bacteria to human, it uses the same genetic code as in this table, okay, for protein synthesis. Okay, so how about reading the frame? Oops. I'm trying to, yeah. Okay. To reliably get from an mRNA to a protein, we need one more concept, that is reading frame. Okay, reading frame determines how the mRNA sequence is divided up into codons during translation. That's a pretty abstract concept, so let's look at an example to understand it better. The mRNA here can encode 3 totally different proteins depending on the frame in which it is read. Okay, these are the frames. So how does a cell know which one of these protein to make? Okay, the start codon is the key signal because translation begins at the start codon and continues in successive groups of 3. The position of the start codon ensures that the mRNA is read in the correct frame. Okay, an example above, an example over here in frame 3. See, you start from the start codon. Okay. All right. So mutations change in DNA. Mutations, the changes in DNA that insert or delete 1 or 2 nucleotides can change the reading frame, causing an incorrect protein to be produced downstream of the mutation site. Okay, you see over here. For example, there is a mutation over here on this. Once there is a mutation, the change in this, it goes like AC, UA, AC, but UA is changed over here. So you see how the forthcoming set of amino acids are changed in here. It was like Vol Pro tire, but it changed to ALA, LEU, and LEU. It goes like this. All right. So this is how the mutation changes the production of proteins. That's, if there is a mutation, then you're not you're producing the required protein. So you're producing other proteins that might also even cause some damages to the organism. Okay, so uh, how was this the genetic code is discovered? In the story of how the genetic code was discovered is a pretty cool and epic one, and I sketched out our version in the pop-up below, so as to not distract if you are in a hurry. However, if you have some time, it's definitely interesting to read that history. Okay.